In our last part of Chapter 3, we're going to take a look at some more of these microorganisms that are out there and start to go into some of these defining characteristics that have been used with this taxonomic classification. And we'll end up just taking a look at some of the different things that you can get into outside of just looking at things like bacteria. And it's really a wide encompassing field of things that you can do out there related to microbiology. So if we take a look at this first idea of the domains of life that we were exposed to in the previous little video there with our taxonomic categories, remember your domains are going to be on the top. And when we look at the microorganisms that are out there, we can find them basically anywhere on the planet, as long as you have some water and a good energy source. And when we consider this idea, the domain archaea actually has not been found to harbor any pathogenic bacteria that cause diseases in us. That really resides with the domain bacteria and some of the organisms that are out here in this domain eukarya. But what we do want to take a look at is some of these representative examples and see why we put these things into these different domains here. And for that, we want to identify some of the key differences between our prokaryotic cells and our eukaryotic cells. And we'll be looking a lot more at the structure of cells in Chapter 3. And then Chapters 4 and 5 go into a lot more detail about various different groups of organisms that are within these different domains that are out there. So this begs our first question, which is, what are sort of the defining characteristics to distinguish our prokaryotic cells from the eukaryotic cells that are out there? And if you look at this idea of prokaryotic and go back to the root of the words, the pro really translates as before, and the karyotic refers to a Greek word for like a kernel of corn. And what people found is some cells that are out there have something that stains to look like they have a kernel inside of them, and other cells that are out there do not have that kernel. And that's one of these defining characteristics of our prokaryotic cells, is that they are before the creation of that kernel, and what people were more than likely looking at is the nucleus. And so when we look at this defining characteristic of prokaryotic cells, not only do they lack a nucleus, they also lack these membrane-bound structures that are found inside of cells. So again, this idea of prokaryotic is before you get these staining bodies inside of the cell itself right there. And again, we can see our domain bacteria and our domain archaea contain these prokaryotic cells that lack these little organs inside the fluid of the cell right there. Their organs are basically floating around without having a protective coating cover on them. Some of the other things that we can find with prokaryotic cells is they reproduce asexually, so they are actually clones of each other. And typically, they're about 10 times smaller than your small kinds of eukaryotic cells that are out there. And when we look at the domain bacteria and archaea, they can all exist as single-celled life, whereas in the eukaryotic domain, only some of them are going to exist as single cells. We do see some interesting shapes for different kinds of bacteria that are out there for our prokaryotic cells. They have some common shapes that we can see displayed right here. If you are coccus, comes from the Greek word for berry, so think like a blueberry. It's a spherical kind of cell. If you are a bacillus, then you're going to be a long rod-like, straight kind of cell here. And people couldn't decide between are you spherical or are you a rod-like cell, so they put the two terms together and we get coccobacillus here. You can also find bent rods or vibrio, and this is something that we see with bacteria that causes diseases like cholera. That's Vibrio cholerae. And then you've got your two types of spiral-shaped bacteria, either a loose spiral, your spirillum, or a tight spiral, and now you're what's known as a spirochete. And one thing I will point out is determining the shape of the cells that are out there is not always easy with our normal kind of light microscope we use in lab, because these cells are microscopic. They are not small, they are microscopic. They are smaller than small. When we look in our domain eukarya, on the other hand, we do find that these cells all contain these membrane-bound organelles like your nucleus that stores the DNA in the chromosomes for the cells. Um, Typically, these cells are going to be able to use asexual reproduction by the process of mitosis, and they will use sexual reproduction via the process of meiosis. And remember, asexual reproduction, you make clones, and with sexual reproduction, you're going to end up with mutations in the children. So the children are genetically distinct from the parents that are out there. Um, and when we look within this domain eukarya, we can find that there are 4 different kingdoms of organisms that fall into that domain. You've got your fungi, including things like yeast and molds out here. Your yeast can definitely exist as single cells right there, and the molds will start to form multicellular kind of aggregates that we've seen in our kitchen. The protist kingdom over here includes 2 very disparate types of organisms, the protozoans, which are single-celled animal-like organisms, and then the photosynthetic single-celled and multicellular forms of algae that are out there. And we'll see when we look in the eukaryotic chapter 5 that there is some work currently being done to distinguish the protozoans and the algae and possibly add a whole bunch of new kingdoms to our taxonomic hierarchy for biological organisms. We can also find our plants. The multicellular organisms that make their food are in this domain eukarya. And then last but not least, the multicellular organisms that ingest their food are the animals. And this will play a role for us because we do take a look at some of the microscopic kinds of worms that are out there that are definitely included in the study of microbiology. There are some other non-cellular kinds of microorganisms that are out there, and these would be things like infectious particles, so things like virions, viruses, and prions. When you look at a virus, it's basically a nucleic acid with a protective coating that could just be protein, or it could also be a membrane kind of structure on the outside. And here you can see a picture of HIV that's actually budding off the outside of its host cell. And viruses are not cellular because, one, they don't have those organelles that we see in our eukaryotic cells, but also they don't have metabolism. They aren't actively doing things unless they're inside of the host cell. So these little ones that are budding off on the outside here are basically dead material that waits for a host cell to come along that's going to trigger that virus to go through a series of events, kind of like a mousetrap flipping over there. It just sits there until the proper stimulus happens. And there was a lot of doubt about the presence of these virus particles because you can't see them. They are too small to be resolved with a normal kind of light microscope that's out there. And prions, well, those are just protein particles that can actually cause infections and diseases. And when we look at this idea of size, you can see our eukaryotic cell size range is over here on this side. So we're looking at the larger kinds of cells, and there's a big range in terms of cell size here for these. We can see things with the naked eye down to about this region here in the millimeter size range. When we get down to looking at bacteria cells and some of the things that are found, the organelles inside of our eukaryotic cells, now we're down in what's known as the micrometer range, so 1,000 times smaller than this millimeter range. And when we get to the viruses, remember these are going to be inside of these cells, so they're even smaller still. We're down in this nanometer size range where you need an electron microscope to be able to actually visualize these things. So we want to end up sort of, why are we studying microbiology? One thing is really to understand how these microbes do affect our health. And there are probiotic bacteria that are out there that can definitely help to prevent diseases from occurring. And there are also different kinds of microbes that could potentially, say you used a virus that infects a pathogenic bacteria cell and kills it, that could definitely be something that could be used in lieu of an antibiotic. We want to understand what's going on in the microbes and these kinds of things that might be happening to all the organisms on the planet. And microbes are extremely important for recycling various chemicals that are out there, but also they play a big role as decomposers. They're going to recycle that dead organic material back into the soil and effectively fertilize it. We also want to sort of understand how microbes can be used for our benefit. So I talked a little bit about this idea of fermentation. Think about antibiotics are coming from bacteria and fungi that are out there. And there's also this really interesting science out there known as bioremediation, where they're actually using different kinds of microbes to clean up chemicals in the environment. Things like oil spills can be broken down by different kinds of microbes that are out there. And this holds some really good potential for doing stuff a lot cheaper by letting microbes either naturally or we can tailor them to do specific tasks for us. And if we take a look at the fields of microbiology, you can see there's a lot of different things that would fall under this idea of microbiology, from the study of bacteria to fungi to protozoa to viruses that are out there to worms. And then you can see there's a lot more of really esoteric kind of things that are out there, to looking at studying stuff at the small level here, to agricultural microbiology, because again, these decomposers are providing fertilizer, food microbiology, and study how we can use microbes for fermentation, but also to see about how they can spoil food and cause illness that's out there. So there's a whole bunch of different kinds of fields to microbiology. But for a lot of you, I know this fourth one will come into play because it's required for nursing school. But I hope you will pick up some of the messages, if anything, this idea of sterile technique and the ubiquitous nature of the microbes, that they are everywhere. They're on you right now, they're inside of you, they're floating around in the air, they're on surfaces, and hopefully you will come to appreciate that, because if you do go on to become a nurse, understanding that nurses are one of the primary ways that infectious diseases can be transmitted between patients and throughout the hospital is hopefully something that you will come to appreciate.